Thank you all for coming today. It's very exciting for me to present some of our data from ManyFishes one. So yeah, my name is Laurent Prétôt, and I'm here in my capacity as a co-founder and co-lead of the ManyFishes project, one of those ManyX projects. And today I'm going to discuss the current state of our network, and I will present some exciting preliminary results from our first empirical study, ManyFishes 1, on inhibitory control. And before I get into that, I want to emphasize, that none of the work that I'm going to show today will be possible without a huge collective effort and an amazing team of collaborators. Some of them are here today to cheer me up, and I really appreciate that. And there's a really non-exhaustive list here of contributors, but I really thank everyone for their collaboration. And I'm super pleased. I'm so proud of where we started as a network, and what we've become and what we have achieved since then. All right, so why a ManyFishes group? Well, fishes are among the most diverse group of vertebrates on the planet. They account for nearly 50% of all extant vertebrates, and they also have successfully adapted to inhabit virtually all possible aquatic environments. And so this remarkable diversity in morphology, in ecology, and a habitat that make them, an ideal group to explore how those different factors can shape cognitive abilities. Now, despite the increased number of studies and the discoveries on fish cognitive capacities in recent decades, mostly the past 20 years, there are still several experimental limitations that continue to kind of hinder our full understanding of fish cognitive potential. And those include the use of small homogeneous samples, and also a lack of standardized testing procedures, especially in comparative context. And so that make obviously cross-species comparison even more challenging. And so to begin to address those issues, we established a ManyFishes consortium. It's the first officially big team science project that's dedicated to fish cognition and behavior studies. And that is a collaboration that was established, that started back in 2022, following conversation, mostly informal, among a small group of researchers with diverse backgrounds in ichthyology and expertise in ichthyology, and then was officially introduced at the first Big Team Science conference in 2022, that October. And since then, the network has been holding regular meetings, has expanded its membership. Now we are counting over 30 collaborators from more than 20 institutions across the globe, and 20 countries represented. So that's all very exciting. You can see obviously from the map here. Now, talking a little bit about our network infrastructure, our project infrastructure consists of a website for general information. You can actually scan this code here and access the website directly. We also have an email address, a group email address. This is mostly for external communication and inquiry from people outside the network. This is the manyfishes@gmail.com. We also have a mailing list for the network and project updates that are shared with members. Mostly when they are registering and joining the network, we will give them access to that mailing list. And we also have a Slack workspace for more like instant messaging and group chats. Now, the project has already developed its own code of conduct. So again, that is to prevent misconduct, to promote equity, diversity, inclusion goals. And we have also established a collaboration agreement, and that collaboration agreement lays out some kind of clear guidelines for communication, things like attribution to of authorship, data sharing, respectful collaboration, and it's obviously a dynamic document, so aims to to change with the needs of the network. To promote accessibility and transparency across the network, we currently use a Google Drive repository. So the Google Drive repository includes a Word doc that is, study materials, protocols, manuscripts that we worked on together. We also have Google Sheets for like data entry and maintenance. We have Google Form, surveys, registration. We have Google Slides for presentation, but also FAQ question for projects, and we also share videos that relate to like demonstrations and pictures of the different studies that we're working on. And finally, and not the least, partnerships are an integral component of the network infrastructure, and we are lucky and are honored to have recently become an official partner network on SSHRC partnership grant, building a network of networks in the big team science, and that is the Connect initiative. And we've done that along with other BTS groups. And so those collaboration, allow us to receive a fund that we can use towards supporting research activities within the network, mostly student activities, and that is that support is obviously essential for the viability and the growth of the network. Now I want to discuss a little bit about the main goals of ManyFishes. They're pretty straightforward, but they are aimed to increase both the number and the diversity of fish samples that are used in comparative cognition research. And so to achieve this goal, we aimed to facilitate collaboration across researchers and institutions, and so develop standardized yet suitably adapted procedures for wide application across species and settings. And so that refers to the principle of species fair approach and functional equivalence. And finally, promoting the use of open science tools in fish behavior studies. And so by fostering cross-disciplinary and dynamic collaboration, we enable the investigation of key research questions in the field of fish cognition, and we do so from multiple perspectives. And so that offers, a more comprehensive understanding of the breadth of fishes' behavior and cognition. All right, so now I'm going to talk about ManyFishes 1. So that is our first empirical study of the network. It focuses on inhibitory controls. So inhibitory control is the ability to suppress those automatic, impulsive, or habitual responses in favor of more appropriate actions. Inhibitory control is a central component of executive functions, and that's involved in a wide range of critical decision-making behaviors in fishes and other animals. And it's often considered to be highly correlated with general intelligence and cognitive competence as well. Now, the current literature on inhibitory control seems to indicate that fishes saw at least some level of similarities, both at the performance but also at the mechanism level with other vertebrate groups, including mammals and birds. But some of the current research on inhibitory control in fishes so far is almost exclusively confined to like single laboratory or groups that use small samples and single or just a few species. And so that preclude those like wide-ranging comparisons and also interpretation of existing evidence. So what are the goals of ManyFishes 1? Well, there are twofold. So the first one is we want to to build a large species diverse data set to better understand the evolution of inhibitory control across fishes. And second, we want to demonstrate the functionality and the feasibility of ManyFishes project. And so that will work as a proof of concept. And that's an approach that has been used by other ManyX networks. ManyFishes use a standardized version of a widely used paradigm that is called the cylinder task. And so in the task, and you can see the video at the bottom here, in the task, subjects have to detour a transparent cylinder. You can see maybe the cylinder here and that fish detouring it to get the food from it. And that is considered, in that case here on the left, a success because the fish has successfully detour the cylinder without bumping into the cylinder or touching it to get the food. So that would be considered a success. Now, a failure in the task will look more like what you see here on the right with the fish bumping into the cylinder. And this one is really working hard and even end up moving the cylinder by itself. And so what makes the cylinder task a great choice for ManyFishes? One is from a methodological perspective, is that the task is relatively simple to implement. I mean, it requires a cylinder. It is minimally invasive. It's also a versatile procedure that's been used to measure diverse cognitive skills across a wide range of animals so far. So there is quite a lot of literature on the topic, and that includes fishes. So that makes it suitable for a big team comparative research project. Now, another more conceptual reason that makes ManyFishes one and the use of cylinder task as an interesting approach is that a previous large comparative study using the cylinder task with mammals and birds, and that was a study by McLean et al. In 2014, they found a positive correlation between a species' inhibitory performance and their absolute brain size, and it was the volume. And you have actually data from that McLean study shown here for birds and mammals. But more recent adaptations of the cylinder task found that guppies, and here highlighted in red, and also sticklebacks, so two fish species, actually achieve levels of performance that are similar to, if not higher, than some of those mammal and bird species. And so together, those data speak against the generality of a large brain, high inhibitory control association, as was previously proposed. And so ManyFishes one provide that unique opportunity to fill in the gap, I would say, in the literature of vertebrate inhibitory control, and doing that more systematically and more widely. So I'm now going to show you some preliminary results from our study, and I want to emphasize those analyses are not finalized just yet, but I'm pretty excited to show you what we got so far. I also want to say a big thank you to Tyrone Lucon-Chicato and Elia Gatto, University of Ferrara, for helping with those preliminary analysis and some of the visuals that I will show you today. So some general information on the study so far. So we have tested until now 440 fishes subjects from a total of 22 species of fish. Those species are from freshwater and marine environments. They are all teleost fish. So teleost are by far the largest group of ray-finned fishes. They make up about 96% of all extant species of fishes. Now, as you can see from this graph here that shows you the species on the x-axis and the proportion of success across trials in the task, the top number on each bar here is the counts, the trials run per species. And so as you can see from this graph, we find substantial interspecific differences. And so that's a first really important finding from a comparative perspective, because it implies that inhibitory control, among other cognitive traits, may have evolved differently across the fish lineage. And so those kind of results open new research questions regarding factors that affect performance in the task. And I'm going to discuss that in a moment. Now, this is another view of those data. When we are feeding species as a random effect in our models, we find significant interspecific differences in performance, as you can see from zebrafish here to Carassius auratus. And so that figure here shows how each species intercept differs from that mean intercept that's located here in the middle. So it's roughly corresponding to variation across species, and that also take into account other model predictors. So here, as you are moving toward the right, those higher order ratios are also indicative of better performance in the task. Now, a second interesting finding is that the species random effect that I just showed you explain a relatively low proportion of the variance in our sample. But the variance explained by the individual random effect, and those are the data that you can see here. So again, within each species, indicate like a much larger presence of individual variation. So clearly we have both interspecific and also inter-individual variation within species. And you can see here some of those species have those very large variation, while others are much more narrow. Now, a third finding, and again, quite interesting, when we are examining how history influence performance, and by history, we have tested species that were either wild caught or captive bred in the laboratory. And when we also examine age, and so we have tested three categories, adult, subadult, and juvenile across our sample. We find that both factors significantly predicted performance as fixed effects. And so you can see here on top, in terms of history, with the wild caught animals. And you can also see here the data for the age. So again, juvenile and subadult as compared to adults. And that's an important finding because it suggests that history and age have an effect on performance estimation, and that should be considered carefully when comparing data from different studies, for example. Now, what was also interesting and a little bit surprising is, well, sex, and here sex, we have tested female, male, and we also had an unknown category. This one only marginally predicted performance, and trial, as you can see here, aligning almost perfectly with the average, did not predict performance. Now, trial did not predict performance, but we found a significant interaction between species and trial. And that is, as you can see here, the outcome on the y-axis across trials. We have some species, a few species that learned the task, improved performance over time, like this one here, while others did not. And we see those pretty flat data set here. And so those results are interesting because they suggest, that different species might use or exploit different strategies or maybe different cognitive functions to deal with the same task. And that could be, hypothetically, due to maybe differences in socio-ecological factors, among others. And for my last set of data here, I want to say that, well, ManyFishes was also a great opportunity to look into possible effect of location or laboratory. And our sample actually had the chance to have one species, guppy, that we tested in 3 laboratories. We had one in Macquarie University in Australia, one in Stockholm University in Sweden, and one at University of Ferrara in Italy. And interestingly, as you can see here across those graphs, we find that using the same species, fish performance significantly differed across those laboratories. And of course, that suggests that other factors like strain or maybe husbandry, or it could be also an experimental error or bias could affect our results. So what are the next step for ManyFishes? Well, we are planning on conducting some phylogenetic analysis to investigate if there is an evolutionary trend, and also help us reconstruct the evolutionary history of the trait. And we're particularly interested in whether species more closely related also perform more similarly in the task. And so to achieve this, we're going to create a phylogenetic map, phylogenetic tree to be used for like lambda estimation. So that's going to quantify the phylogenetic signal in a trait by scaling internal branch length. And so for that, we already have access to pretty robust phylogenetic maps of fishes, but it's about now to find the most compatible map. And as I discussed earlier, we're also interested in comparing the performance of fishes to the performance of other vertebrate groups that have been tested in the task before. And that will help us kind of fill in the gap in the inhibitory controlled vertebrate literature. And finally, we plan to conduct additional analysis that could be also part of spin-off projects, because there's so much that we can obviously investigate whether trophic level or dietary breadth affect performance, whether sociality, and that can be also parental care influence performance. We're also obviously interested in various brain measures and how those might predict performance. And finally, that interesting question about visual acuity, but that's obviously something that is general to other species and other taxa as well. All right, so that's the end of my talk. I hope I was on time here. I want to thank you all for your attention. I also want to take this opportunity to thank all those networks that are on that slide here, partners, for their support. And I'm happy to answer any question if we still have some time.
Hello. Can you hear me all right?
Yes, hi, Erin.
Hi. Great talk. Exciting work. I'm curious. I know nothing about fish, and um, it was counterintuitive to me that the wild caught fish performed better in this. And so I'm wondering if that's a common difference in the fish literature and why that might be.
Yeah, that's a fantastic question. So something that obviously we don't know about wild caught fish is their experience, right? Their life experience. That's something that we cannot track easily, because when they are wild caught, you don't know much about their history. So there's clearly they could be clearly an effect of experience, and I'm saying general experience and with your environment, and so maybe the richness of the environment is going to shape, those cognitive abilities and train you to be better in those kind of tasks. Um, another aspect of it could be that wild caught fish work for survival in the wild, right? So solving the task or maybe higher performance could be due to the fact that it might be more meaningful or more important to them. Um, but yes, some studies show in the literature, in the fish literature, that wild caught sometimes outperform captive bred species, while the other way around is also possible and exist. And we have actually reviewed some of that work in a literature review that we are currently that's currently in review actually. So
Very cool.
question.
Thanks again for the talk.
And if I have any of my fish friends here wanting to chime in, please.
Thank you so much again, Laurent, for such a wonderful talk. Does anyone have any other questions? If not, I think we could end it there. If you do, you could just type them into the chat or even just unmute yourself.
Thank you all so much. Thank Christian for making it, for your support. That was great. And yeah, looking forward to see those results somewhere. Thank you, Kyra. Have a great day, all.
Thank you all for coming.